I'm going to start out by showing you some of the recent, and in fact, this is today's map. And you can see that the total number of confirmed cases worldwide is 441,000 plus, 19,784 deaths, and there's 111,933 totally recovered. Now you have to look at that number and realize that that number is very low, that last number, because it does not include people who were identified as having the disease but were not hospitalized. So those are people that were released from hospital or clinical care. As you can see in the map, the United States there on the right is really becoming one of the epicenters, as is Europe. And I'm a little surprised that Africa and more South America hasn't, but they probably are gonna get their sort of things too. Now, on the next slide, I just added this today because you might find it, the URL is on the bottom, but you might find this helpful. If you go to the actual site, you can click on the different myths and they'll give you a little bit more information to the extent that you believe FEMA. I went through it and most of it seems pretty accurate. So that's a site that you can go to differentiate between a lot of what's out there that may or may not be true and at least something that seems to have some accuracy to it. Okay, now there's some questions that everybody's asking, but those of us that work in the area are asking much more profoundly and in more detail. What's the original source of the virus? And by the way, there's a lot of text here because I'm going to share these with my students, and because we're doing this virtually, I want them to have more text to study from. So there's a lot more text on these slides than I would normally put on the slide. So I'm apologizing for that. How did it get into the human population? Could it come from a biological research or a bioweapons lab? How does it spread from person to person? How contagious is it? Are there super spreaders? Now, in the case of MIIS, the one out of the Middle East, there was a guy that went from Saudi Arabia to Korea. He was a worker in Saudi Arabia, went home, developed the disease, went into the hospital, and within about a week, 250 people in the hospital, or plus or minus a few, had the disease, and it spread through the ventilation system. And MIIS is not typically that contagious. So these sorts of things happen every so often, and we don't really understand why. What's the fatality rate? And you'll see why it's kind of hard to measure that. What's the range of severity? You've probably heard a lot about that. Who's most susceptible, and can someone carry it and be asymptomatic? And can someone who's asymptomatic spread the disease? And what's the reproductive number of the virus? And that's actually very critical, and how that can be modified. What's the best way to contain the spread? Certainly not the way we're doing it now. It's possible to develop an effective vaccine, but how long is it going to take? How does one test its efficacy? You can't very well vaccinate people like we did in previous centuries, where you vaccinate orphans and prisoners and then challenge them with a disease. We're not allowed to do that anymore, thank goodness. And then what I want to do is actually present some more of the epidemiological data, because that's how we get a handle on a lot of what's going on. First, I want to cover what this disease does to the body. We do know that it can cast a storm over the whole body, and in fact, in the most severe cases, that's what's happening. Coronaviruses, there's not that many in humans that are known, but most of them cause minor diseases like common colds. But in this particular case, it can really spark a dramatic response by the body to the infection. In fact, in China, the diagnosis is based not only on the PCR assays that are being done now, but also on looking at a pattern of in the chest by doing chest scans, where they actually see holes sort of knocked in the lungs in more severe cases. But if you look at the diagram on the right, of course, there's all kinds of things going on. Some are direct, some are indirect. Respiratory ones on the right there: coughing, sneezing, runny nose, shortness of breath, breathing difficulty, sore throat. In the severe cases, pneumonia, severe acute respiratory syndrome in lungs with inflammation and congestion. And in extreme cases, you can have kidney failure, although that's usually secondary to other things that are going on in the body. But you're going to see decrease in white blood cells, cardiovascular damage in severe cases, diarrhea sometimes, although not so much with this one. And then, of course, fever and fatigue. For most patients, the virus begins and ends in the lungs. Actually, it begins in the upper sinuses and then spreads to the lungs. The reason it's so contagious is that it spreads in your upper sinuses, where it can then be dispersed rather quickly. The attack appears to occur in 3 phases: of viral replication, where it infects and then starts to make new virus. Then your immune hyperactivity, and that's one of the critical problems with this disease. And then pulmonary destruction. Now, this doesn't happen in all the patients. Only about 25% of the patients get to the point of respiratory failure. It causes minor symptoms in about 82% of the cases, and then the remainder are severe or critical. During the early phases, it rapidly invades 2 different classes of lung cells. There are cells that make mucus, and they protect the lung tissue from pathogens and help maintain tissue moisture. And there are cells that contain cilia. They move the mucus, clearing out debris and that sort of thing. And they work together to give you the protection you have. It likely infects and kills the cilia cells first, which then slough off and fill the patient's airways with debris and fluids. The immune system then floods the lungs with immune cells to clear the damage and repair the lung damage. And then sometimes this gets unregulated, goes out of control, and it ends up damaging not only clearing the damage that was done, but causing more damage in itself. And during the next phase, the lung damage continues to build, and this can lead to respiratory failure. Even if the patient survives, they're often going to have lung damage permanently. And again, as I said before, it sort of punches holes in the lungs, giving a sort of honeycomb appearance in these chest cans, and this has been used by the Chinese to help identify the disease. The holes that occur are caused by the patient's immune system. It creates scars and stiffens the lungs, and this is when you have to put somebody on a respirator, and sometimes that's the end of it. In severe cases, the lungs are flooded with fluids, and the patient can't breathe, leading to death from that. Basically, the alveoli and blood vessels area, the permeable area where the gas exchange occurs, fills with fluids, and that's often the end of the patient, unfortunately. The stomach is interesting. SARS, the original SARS that we talked about, 2002, 2003, and MERS, nearly 1/4 of the patients had diarrhea, and it seems to be caused because the virus infected cells in the intestinal system. What's interesting is this virus invades using the same sites as the original SARS and MERS did, and yet there haven't been many cases of intestinal problems. And so it seems like it would bind to the same receptors on those cells, but it, for some reason, doesn't seem to be doing that. It's very important to know whether this is spread by feces, because then you've got this fecal-anal issue related to fecal food contamination, that sort of thing. Right now, it doesn't look like that, or if it is, it's limited. And I'll get to that later, but the interesting thing is that people say, well, I've seen virus here, I've seen it in my pets, I've seen it this. The assays that are used don't detect virus, they detect viral RNA, and that could be from dead viruses, live viruses, or just free RNA that's floating around. So you have to be cautious when you see these reports that, oh, I detected it here, I detected it there. If they're using a detection that does not actually detect live virus, then you have to be careful how they interpret the data. The blood, of course, the virus doesn't seem to get into the blood, but there's a whole bunch of things going on because of your immune system response to it that cause all kinds of indirect changes in the blood, elevated liver enzymes, low white blood and cell and platelet count, low blood pressure, rarely, but sometimes acute kidney injury and cardiac arrest. But it's mostly because of something that we call a cytokine storm. It's an overreaction of your immune system to the infection. And you may have noticed that they've used a couple of drugs that they said were somewhat helpful in treating the disease in seriously ill people. One of those drugs is a drug that's used to treat malaria, and its primary function is to reduce the response of the immune system. And so that the point is that if they can reduce this reaction of the body, you might be able to save patients that way. Again, cytokine storms create inflammation that causes fluid to seep from the lungs into the circulatory system, and then you might get issues that way. It's highly unlikely to find the virus in the blood, except in very severe cases, however. So the blood system, even though people are starting to use blood serum as a treatment for the disease in severe cases, because people who have recovered will have had an immune response that provides some protection, actually a lot of protection, and they're trying to use that to treat some of the patients. But there is not virus in the blood. It's your own antibodies and such that are a result of your immune response. People at first thought there was a problem with unborn children being infected, but as far as we can tell, that's not happening. It did not happen in SARS or MERS. There were 2 early reports that it did, but in fact, they've looked at 9 infants who were delivered from COVID-19 infected people or women, and none of them were infected with the virus. Okay, an epidemiological study can provide some of the answers to what we need to know to deal with this. And I'm going to give you a little, just a real introductory course in epidemiology here. There's something called the reproductive rate, or R0. You've heard of that, I'm sure, by now. It's used to measure the transmission potential of a disease, and it's thought that as a number of secondary infections produced by a primary person. Not secondary infections by secondary people, but only the primary person. So for example, if you have R0 for measles in a population is 15, which would be pretty high for an R0, then you'd expect it to spread rapidly, because each new case with measles would produce 15 new secondary cases, assuming susceptibility to the disease. Now, this R0 is affected by several factors that some we can control and some we cannot. The rate of contacts in the host population, that's what we're trying to control right now. The probability of infection being transmitted during that contact. Duration of the infectiousness of somebody who's sick, and in general, for an epidemic to occur in a susceptible population, R0 has to be greater than 1, in which case that means the number of cases is increasing. Of course, often not all contacts are susceptible, that some are immune because they've either had the disease and have been conferred lifelong immunity because of that, or they've had some sort of immunization or whatever. So not all the people that are in contact with somebody who's infectious may pick up the disease. In general, the larger the value of the R, the harder it is to control an epidemic. Again, hardly ever do you find a population that's susceptible to an infection. Everybody's completely susceptible. There's always some people that are not susceptible. And so there's something called R0X, which is your basic reproductive rate discounted by the fraction of host population that is susceptible. So for example, if flu, they got, let's say you have an R0 of 12, which would be very high for flu, it's more like 2. But in a population where half of the people are immune because, let's say, they were vaccinated or whatever, then you multiply that by that factor. In this case, it'd be 0.5. So the R0X is 6. So the idea is to try to reduce the R0 in whatever way you can. And right now, since we don't have vaccines, it's happening naturally, because as people get sick and then recover, they're going to be resistant to reinfection. And there's lots of evidence for that now. And that's normal. Actually, for most illnesses, that's quite normal. And then we have the social distancing, which is another way to handle some of it. There's something called herd immunity that occurs when you get a significant portion of the population has been vaccinated or otherwise exposed to the disease and becomes immune. And basically what this does is as people that are infectious circulate in the population, there's going to be fewer and fewer susceptible people that they interact with as the number of people that are resistant increases. At some point, it's as low as 60% in some cases, more like 70 or 80% in a lot of cases. It gets to a situation where the likelihood of somebody who's contagious and spreading the virus coming into contact with somebody who's susceptible is reduced to a level where it just doesn't spread, and the effective R0 is below 1. And then at 1, it stays in the population, but doesn't spread. And once you get it below 1, it'll eventually drop out, and the illness, the pathogen will disappear from the population. And that's something we're trying to get to. Just the phone's ringing, and I don't want that to happen. The other way we look at this, and the way you've seen, in fact, I showed you a curve already, is to look at what we call epidemic curves. And it's really a simple curve to generate if you have the appropriate data, but the hard part is getting the appropriate data. It's basically a plot of time versus the number of incidents on a day-to-day basis. It can be then, once you have that information, if you have an accurate one, you can then propose hypotheses about the disease, how it's being transmitted, so on and so forth. It's frequently referred to as an epicurve. And by constructing and examining an accurate epicurve, you can determine, first of all, if there is an outbreak. Well, that's obvious that we have one. If so, when did it begin? Has it peaked? If so, when was the peak? What was the source? Is there one source or multiple sources for exposure of cases? Is person-to-person transmission occurring? And have attempts to control the outbreak coincided with the decrease in the occurrence of the disease? And that's incredibly important, because you need to know whether the methods you're using are having any impact. So the curve I'm showing you here is from yesterday, I believe. I think this is the 23rd, yes. And as you can see, the first cases were in the Western Pacific, which would be well, China primarily. And that peak in the middle of that first group is artificial. They changed the way they were reporting cases at that point. You can then look at the Africa cases, which are thankfully very, very low so far. And then you can look at the European cases, which are a little bit ahead of us. And you can see how they've peaked and they're still going up. We have Eastern Mediterranean, where it's not quite so bad. Populations are smaller. Now you can look at the Americas there, and you can see where it's really starting to take off in the United States and Canada, and to some extent Mexico. This is the cumulative total number of COVID-19 cases in the United States by as of this morning. And as you can see, the case number was very, very low. And it has just started to climb drastically. It's basically on an exponential curve at this point. Now, here's another way of looking at the same sort of information. And this should alarm you a little bit. If it doesn't, then maybe I need to just describe the curve a little bit better. You can look at Japan down near the bottom. They've done a very good job of controlling it. The countries that have done the best job of controlling it are China. And the reason their numbers were so high is because they had to get a handle around it in the first place, and South Korea. South Korea has extensive testing, and they haven't actually quarantined a lot of people. I mean, they've quarantined people. They haven't actually done total shutdowns, shelter in place, that sort of thing. But they're doing it because they have done extensive testing. And that seems to work as well as anything that's been done so far. You can see Iran in there, and Italy, of course, is on a very high projection. And if you look at the US right now, it's climbing, the slope is faster than anything else out there. So this is, again, as of this morning. Problems with models is that you have to have accurate information. As I said before, the Chinese government changed its method of reporting the cases, so that messes things up. And we also found out just the other day that the Chinese have not included people whose test was positive for the virus by the PCR test, but who do not show symptoms of the disease. Well, that means they're underreporting a very large number of people with mild cases that, for whatever reason, weren't counted. On February 10th is when that spike occurred, and it really messed up all the graphs and such, because we really didn't know what was going on. But the number of mild cases, those that don't require hospitalization, have to be counted, because if they're underreported, you don't know how well or how extensive the virus is moving through the population via these people who are not so sick that they are hospitalized. Usually you use simple math to figure out the case fatality rate. You just take the total number of deaths and divide it by the total number of cases, but you have to get an answer that's accurate. Now, getting the death numbers is reasonably easy, not as easy as you might think in some places, but getting the actual number of cases is very hard. So SARS 2002, 2003, the fatality rate initially was thought to be 2 to 3%. It turned out later it was 17% when they had more numbers. Several groups are right now trying to determine the fatality rate. Now the way, if you have different people approaching it from different means, then you can maybe get a good idea of what the fatality rate is. But if you can't count the number of very minor symptoms or cases with minor symptoms out there, it's very, very hard to get the fatality rate. Of course, you want to know what the number of people is that, the number that might likely die or who are going to be seriously sick, because you have to prepare for that. And that's what's happening in the United States right now. You can get better estimates if you know that number for number of isolation beds, equipment, personnel, and media needed. Now, what's happening in New York right now, and actually in Italy and other places, when you don't have the gear and the equipment and the people, they start to triage the patients, and they work mostly on those that are most likely to survive, which means that people that might be very seriously ill but might have made it through aren't going to get the treatment they need to make it through, and you're going to have a higher fatality rate. What we need to know, of course, is all of this sort of thing, but the Chinese in January analyzed the first 425 confirmed cases. 65% had never visited a food market, nor been exposed to anyone showing any sort of pneumonia-like symptoms. This implies, among other things, that the possibility of some infected people that don't show symptoms are passing the disease around. And I'm pretty sure that's what's happening. The most pressing concerns are how to establish the lethality of the disease and who is susceptible, getting detailed information on how it spreads, and evaluating the success of control measures. If I was involved heavily with this right now, the first thing I do is I get as many of the people from China as are willing to talk to me and talk to them firsthand about, hey, what's going on and what happened. This idea of a clinical iceberg is something that's how much of this disease is hidden below the surface, out of view, so that people mildly infected people are spreading it. Fleet of invisible carriers sounds ominous, but in fact, if there is an enormous hidden figure, that would mean that the actual death rate or the fatality rate is actually much lower relative to the number of people that actually got the disease. Now, first case, you probably know this, but it occurred was reported in Wuhan in late December 31st, I believe. That city has a population of about 11 million. The virus is called SARS-CoV-2. The disease is called COVID-19. It's infected so far, as of this morning, that we know of, 422,000 plus. It's killed 18,900 plus. That's a 4.5% fatality rate based on those numbers. And there's 111,933 people who have recovered. That means they've been discharged from medical facilities. That the mild cases aren't counted here. So far, it's spread to 195 other countries. Some of you guys probably know how many countries are in the world, but that's a lot of countries. Wuhan, China, and at least 15 other cities were quarantined as China attempted to halt the spread. Now, they can lock down much more effectively than the United States can because of their government ability to do that. About 50 million people were locked down. There have been 15,626 deaths outside of China, and as of the last count I could find, 792 of those are in the US. It's far surpassed the original SARS outbreak of 2002, 2003. The Chinese province where the coronavirus pandemic began will this Wednesday, today, begin allowing more of its, most of its 60 million residents to leave their homes. ending nearly 2 months of lockdown and sending a strong signal that they think they've got it under control. Now, Wuhan, the city hit hardest by this, will remain sealed off until April 8th. So they're going to start public transportation within the city. Now, this does not mean there's free travel within China. Many provinces and cities have made it easier for residents to move around, but essentially shut themselves off from outside sources. They still face the risk of a full resumption, that a full resumption of travel, work, and normal daily activity will renew the spread of the virus. Now, you'll notice in the numbers the Chinese are showing is that the vast majority of the numbers of cases they're reporting daily now are imported cases. That's because now that they've done this sort of wholesale lockdown, they don't want the virus to come back in from other sources. So they are monitoring everybody that comes into the country from outside for the virus, and that's where the majority of their new cases are showing up. There are people traveling from elsewhere in the world back to China. Now in politics, Trump, science, and medicine, you get something like this happening. This individual, of course, has died. It's really sad that on February 7th, he alerted a group of alumni from his medical school about this new virus, but he was silenced by police in Wuhan and forced to sign a letter acknowledging that he was, quote, making false statements, end quote. He was infected by the coronavirus while treating patients, and he died on February 7th at 3 a.m. But that's, you know, I always ask questions when I get these people to say, well, they think they ought to listen to the politicians. Well, I mean, I look at it this way. If you get sick with a disease, whether it's this one or some other disease, are you going to contact your local physician or someone along those lines? Or are you going to call your congressman, senator, president, or whatever? I mean, you go to where the expertise lies, and when you don't do that, you get into trouble. And that's, I think, where we're going in some places, at least in the United States right now. So where did it come from? It appears to be a zoonotic virus. That is, it jumped from an animal species to humans. A lot of human diseases are zoonotic in origin. Coronaviruses circulate in a number of different species. They're RNA viruses. RNA viruses don't have the editing functions that DNA viruses have to keep mutations down, and therefore they introduce a much higher frequency of mutations into their population than you would for something like smallpox or some other DNA-based virus. Occasionally, with a virus with the appropriate genotype, in other words, genetic characteristics, for survival in humans will move from the zoonotic source in one or more steps into humans. It's usually more than one step. Flu does that. It usually goes from birds to swine or something like that. This virus may have jumped from animals to people at a wet market, although the numbers seem like it probably was circulating before then. But it could have come from the wet market at an earlier time too, because based on its genome sequencing of this and other viruses for comparison, and those other viruses being collected from animal species, it looks like that came from bats, and it may have come through another species to get to humans. The genome was sequenced from the very first isolate right away. The Chinese got that really fast. And it was almost an exact match to a genome of a coronavirus collected from a pangolin, which is the mammalian species that seems to be involved. A virologist who helped identify SARS-related viruses in bats in 2017 said that the strain was clearly mammalian virus. And again, you can tell by the sequences, because the sequences tell you something about the proteins, and these proteins tell you about something about how they can get into another organism. But a very recently published study shows that the sequence change that allowed the virus to infect humans is in a region of this S protein, the spike protein gene, that's never been associated with changes that would impact the host species of the virus. So this is a brand new mutation nobody's ever seen before, and nobody would have predicted that a change in that part of the gene would have done this. So when people are trying to intentionally manipulate something, they usually do that based on previous knowledge of what changes would be needed to get the impact they're after. And modeling with the information we had until now would not have suggested that this present structure would allow infection of humans. It just never would have made any sense that it would do that. The other thing we know about it is that well, of course, if you've done this sort of thing, you know that if you intentionally manipulate a genome, it's done with molecular tools and sequences that leave a signature of any sort of manipulation in the genome that's easy to identify if you compare it to other genomes of similar similar viruses. There's no such signature in this genome. So these facts argue against this virus being released by a biotechnology or bioweapons lab. It looks like it's a natural source, and there's no indication whatsoever. If somebody wanted to cover up that sequence manipulation, you could, in most cases, unless you really were good, you probably couldn't do it. Not only that, but if you wanted to do an intentional release of something like this, why would you bother to cover it up? So again, it looks like it occurred in a wholesale market, and there's a lot of live animal sales that are actually illegal in China, but they still go on. And initially when they saw this, I remember the very first ProMed report I saw, it said, well, it can only go from animals to humans, and therefore it's not going to go from human to human. But then they began to see on about January 20th that it was moving from person to person. Again, the coronaviruses were collected from wild pangolins and compared to the sequences from infected patients, and they're 99% identical. You say, well, that's not 100%. Well, it turns out that when you get a virus infection of one of these coronaviruses into a human, what comes out is a cloud of very similar but not identical new viruses. It mutates that fast. Now, the reason that these pangolins are used is like rhino horn. Of course, as far as we know, it has no proven medicinal value, but it's used in traditional Chinese medicine to help with ailments ranging from lactation difficulties to arthritis. And it's a threatened species, but people still sell these in the markets and such. It's an anteater of some sort. I had to look up, I didn't ever even heard of one until this happened, and I had to look up and see what the heck it was. Again, I'm not going to go into detail here, but the genome is 29,700 nucleotides long. That means relatively small virus. It's organized just like all the other coronaviruses out there. What is interesting is that there are several genes in this virus that lack sequence similarity to anything else we know anywhere in all the investigations that have been done worldwide in other species. And so normally the way you figure out the function of a gene is by comparing its sequence to those that have already been identified and characterized in other species. In this particular case, there are no such comparisons to be made because we've never seen the sequences before, so we have no idea what those genes do. Again, you see the first isolate there, by definition, is 100%. Then you had two more isolates, basically from two other patients, and they're 99% already, and then you drop off pretty fast after that. I don't want to get into too much detail on this sort of thing. This you might find interesting. This is how the virus gets into cells. And this is why it's important that we follow the SARS virus spike protein gene carefully, because it's changes in that determine what species it can get into and what types of cells within that species, lung cells or whatever, that can be infected. So the SARS virus attaches to a receptor called the ACE2 receptor on the outside of cells. That ACE2 gene encodes the angiotensin converting enzyme 2, and it's been proven to be the receptor for both SARS-CoV, the original one, and this one. As it attaches to that, that facilitates its infection into the cell, where it then replicates and makes new parts. And those pieces bud off the outside of the cell. You probably heard that you can wash your hands and thoroughly destroy this virus with soaps and such. And that's because as it buds off, it takes a piece of the cell membrane with it. And that becomes an integral part of the virus. And when you expose that to detergents, or in some cases alcohols, it destroys that, and then that destroys the virus. Now, the sequence of the viral spike protein, and in particular the presence of specific amino acids at key positions in that protein, determine whether it can interact with a host's ACE2 sites or not. And that's what had to change for it to be able to go from bats to pangolins to humans. And it was mutations in that allowed it to get into humans. If that doesn't happen, it can't infect humans. Now, the problem for us as humans is that that virus is not well adapted as a human pathogen, and therefore it causes a much more extensive reaction in some humans than others, and therefore a more serious disease. It's a well-adapted pathogen doesn't kill its host. It needs to get passaged around. If it does kill all its hosts, it's going to die out pretty fast. Now, one thing that is worrisome and maybe the reason for some people, elderly people and people with some health issues having problems is that just this morning a report came out that ACE inhibitors and angiotensin receptor blockers, which are heavily used for treatment of certain medical issues, may actually increase the mortality rate in elderly COVID-19 patients. And that's because when you use these blockers, the cells start making more receptors, and so therefore you have more receptors on the outside of cells for the virus to get in. Again, this is just another way of looking at the genetics. You'll see those in red on the right. That's basically, it's called a phylogenetic tree, but think of it as a family tree, and you can see that the beta coronaviruses, that's the group down on the bottom part of the graph, include those, and you'll see there's SARS-CoV is in that group, and the civet cat ones and a bunch of bat ones. They're all very closely related. If you look at just the S protein and just look at the sequence of that, it explains why it gets into humans. Definitely very closely related to those that have been in humans previously. What we need to know we need to get a clear view of the contagion and plug the holes in our understanding of the disease so that we can inform public health decisions. The present model is the new coronavirus is a close cousin of viruses that infected bats that jumped into humans, presumably at a food market, but someplace in China. The infected people unknowingly spread it, setting off today's what's going on right now. We now estimate that it takes about 5 to 6 days, possibly upwards of 14, but most people show symptoms after becoming infected within 5 to 6 days. However, there's been at least a few reports. The longest one I've seen is 39 days. So if this is accurate, then some of the quarantine people might in fact still be spreading the virus, but it's going to be rare. 14 is actually a pretty long number. 5 or 6 is actually normal. What's interesting is few children have shown the disease or been infected, or at least if they're not infected. If they're infected, they're not showing it. Although there's been a couple cases of that in Germany and in China. A study published in the New England Journal of Medicine earlier this month speculated that children might be less likely to become infected, or if infected may show milder symptoms than adults. This is probably good if it doesn't spread in children, because children are not real good at washing their hands and keeping their hands away from their mouths, eyes, and nose and such. And so if it doesn't spread in humans or in children, that's actually good. I'm not going to go into the detail. You know about that it was finally declared an outbreak public health emergency. To put that into perspective, here are the other ones that have been declared since 2005 when this was created. The 2018 Ebola outbreak, not to be confused with the one in Western Africa. This one was in the Republic of the Congo. The Zika outbreak, and then the polio outbreak, or the polio emergency, because they're not getting the vaccinations done in places where it needs to be done. That's more political than it is anything else. And then the 2014 Ebola outbreak in West Africa. So how do we test for this thing? You're going to hear some terms you may not have heard before. They're just means to do DNA or RNA-based tests. The test we use for this is called an RT-PCR assay. It's been available worldwide since January 13th. It was approved for use by the WHO. Now, there's been a lot of misinformation about this. The WHO shipped coronavirus tests to nearly 60 countries at the end of February, but the US was not among them. Why? No discussions occurred between WHO and CDC about the WHO providing the test to the United States. This is consistent with experience since the United States does not ordinarily rely on the WHO for reagents or diagnostic tests because of sufficient domestic capacity. WHO lists 7 different approaches, including that of China, the United States, Japan, Hong Kong, Thailand, France, and Germany, each one targeting different parts of the COVID-19 genetic profile. So they're all targeting the RNA genome, but they're looking at different parts of it. The German assay became the approach WHO circulated as its preferred model. Germany released its protocol on January 17th, but the US decided to have the CDC develop its own. That protocol was published on January 28th. The CDC's test was different and somewhat more complicated than the German test, and it worked in the CDC lab, but when they sent it out to the state labs, it didn't work so well. State labs started developing their own tests, and they were ready to use them, but had to wait for emergency approval from the FDA. All of this added up to the delay in testing capacities, which resulted in fewer Americans being tested. And it turns out testing is critical to know what you really have. Now, it's normal for countries' advanced research capabilities to develop their own tests. It's also unlikely that WHO offered to sell kits to the US because that's not normally what the organization does. Normally, it provides kits to countries that can't afford them or don't have the technology to develop their own, and they do not provide it to more of the developed and richer companies or countries. And the reason for that is because they need them in these other places. So the US did not turn down an offer from WHO to get tests or anything like this. This is normal procedure for any of this sort of thing whenever it happens. The choosing to develop their own tests instead of using the one circulated by WHO, other countries have done the same thing, and I don't know why you would pick. I haven't seen the US test, so I don't know why they think that's an advantage over the German test. German test looks like it's a very simple, I could have developed a test. It's not that hard to deal with and to develop these things. But for whatever reason, the US didn't want to. Now, here's what happened. If you look at these lines, vertical lines, the one on the left is a control. You'll notice that there's 1, 2, 3, 4, 5 dark bands down. Those are the results of the test. And they did tests with 3 mock and 3 actual isolates to test using different genes from the, they're targeting different genes in the test. And for these tests to be accurate, all 6 of those dark fragments ought to be where they are in the control. And this was a early US test. You can see Mach 1 didn't work, Mach 2 didn't work, Mach 3 didn't work, and the 3 actual tests didn't work either. So that was done at a state lab. So obviously there was a problem with the test and they had to revamp it. I'm not going to go through the detail in this in the German test, but they screened for 3 genes, the E gene, the RDRP gene, and the N gene. They're in locations in the genome. And the first line of screening is for the E gene. And then they confirm that. This is all done in one test. They do the 3 all together. And looking for the RDRP gene, and then they look for the N gene. And so they look for the first 2, and then if they don't get it, they do an additional confirmatory test to look for the N gene. And that's a positive. I don't want to go through the reaction here, but that's how it's done. The assay, this is something that there's a real limit and people don't always understand. The assay detects viral RNA, not intact viruses. It doesn't differentiate between RNA from a live virus and that from a dead virus or from free RNA fragments floating around in the sample. It's very difficult to determine whether the RNA came from a live or a dead virus. The methods to do this have been developed for bacteria because you can grow bacteria, just grow out on plates. But because viruses required growth in host cells, to draw them out. There's too many variables introduced in the experiment to produce a reliable answer. A positive test in an individual who has recovered from an infection is not necessarily an indication that that person is still shedding live virus. All it tells you is that there's RNA there from the virus. The initial US tests produced by the US, or approved by the USDA required specialized training and equipment, which is something you don't want if you want people doing lots of tests. Not only that, but some of the supplies required were only available from Germany, a certain company in Germany that provided them, and they are obviously in big demand. Roche has developed a new test that's more simple and quicker to get results. They've already begun production, and my understanding is it's been approved. And they can do up to, with their units, which by the way cost about $250,000 for their instruments, but there are some of them around. There's about 110 of them in the country right now, but they can do up to 4,128 assays a day. And nice thing about their assays is it also puts it into a spreadsheet with other information, and so you get a readout that's very useful. This test also detects RNA. It will not differentiate again between live and dead virus. Cepheid, which is a Bay Area company, on March 23rd, had their test approved, and the machines that run the test are already in some healthcare facilities in the US. Like those tests already in use, this new test looks for bits of the virus RNA in samples. They started shipping tests this week, and Cepheid is well known for they're the gold standard for TB drug resistant and multi-drug resistant testing. There's about 5,000 of the machines in use in the US, but they're going to have to get more of them out. I'd like to brag a little bit about this because the technology spun out of research done at Los Alamos and Lawrence Livermore National Labs in the mid-1990s to develop field tests to rapidly detect human pathogens. And the human pathogens we were looking for might have been found in places like Iraq and places like that, if you get my drift. I developed the assays for those. They detect anthrax, plague, tularemia, influenza, and several other pathogens. The engineers at Lawrence Livermore developed the original instrument. I used to have one of the instruments, but I gave it back when I retired. But this has been around since the mid-90s. Cepheid got the permit or the patents for it and have been working with it for some time. One of the other problems we've had is that there's been there's been an attack on Health and Human Services, a cyber attack, and it was an attack primarily what's called a distributed denial of service attack. So it didn't apparently get into the issue, but this is something we really don't need, is to have people try to shut down or paralyze the systems that are working on this. Again, you know about the imposed travel. I won't go into details there. As you can see, even now the US and Canadian and US and Mexican borders are shut down to all but necessary travel. There's been some problems with the current administration. They move very slowly. Trump, actually in 2018, about the time that he announced the Space Force, he made some derogatory comments when he cut the funding for this kind of work that was put into place during the Ebola outbreak of 2014, so that the US could participate with other countries in developing means to follow and deal with these diseases. And that resulted in the CDC's budget being cut 40-fold for this work, and they went from working with 49 countries down to 10. Now, Trump, speaking in India on the 25th of February, said that while the coronavirus is, quote, very serious thing, end quote, it is, quote, under control, end quote, in the US and will go away. We obviously know that that's an inaccurate statement. I don't want to go into too much detail here, but you need to know something about quarantine, federal and state laws. First of all, the US government can only quarantine people that come in from outside of the country or that cross state lines. And there's only certain diseases that are allowed, that are authorized for quarantine. They are set forward by an executive order. They include cholera, diphtheria, infectious tuberculosis, plague, smallpox, yellow fever, and viral hemorrhagic fevers. SARS, the original SARS. So far, this one has not been added to that list. By statute, U.S. Customs and Coast Guard officers are required to aid in the enforcement of this. What a lot of people don't realize is that federal quarantine authority does not include state authority. In other words, if a governor in a state, the feds can't say they're going to impose a quarantine in California. That has to come from state officials. And the only way the Fed has any control over that is if it crosses state line, it's interstate spread. But if it's in a particular location, for example, let's say San Francisco, the federal government cannot impose a quarantine in San Francisco. The governor of the state would have to do that. Now, how long should we shelter in place? Of course, people are saying 3 weeks is long enough or whatever. Here's a back of the envelope simplistic way of looking at it. Okay, you need to consider the length of the incubation period, 7 to 10 days. 14 days is more reasonable because that's what people have used so far. The length of time that someone is infectious can spread the virus. Most symptomatic infections last 10 days to 2 weeks. More severe cases last 2 to 3 weeks, but those people are hospitalized, so they're not out in the public anyway. The length of time that the virus can survive in the environment without a host on fomites and other surfaces, the longest seen so far is 9 days. So add all those up, you come up with a minimum of 37 days, 5 weeks, 2 days. Add a few days for safety, and let's say 6 weeks. Of course, once you've done this, and let's say you've stopped what's happened, you've stopped the spread, then you've got to stop the reintroduction of the virus, bringing it into the clean locations. And that would be meaning limiting travel among different population centers until the extent of the virus in the host community in different locations is established. Now, are we prepared to quarantine people that haven't sheltered in place from other states or cities? Can we even do this? Is it legal? I don't think so. The Chinese are doing this right now. They check everybody coming in from outside the country for the virus. And if you're gonna, once you knock it down, if you want this to work, you got to pretty much do that. Now, as you know, the Trump administration said yesterday they want to end the shelter in place after only a few weeks, like 3 weeks. That's not going to be long enough. It just isn't long enough to get the numbers that you need. I'm not going to go into quarantine centers, but we only have one in the United States. It's at the University of Nebraska Medical Center. And there are quarantine centers, and then there are infectious disease complicated, you can see what an infectious disease, like a patient isolation would be to keep people from spreading. That's the ideal situation. As you know, we don't have that. I'm not going to go into evacuations. We already know the spread, but there is one thing that has come up. Several people have asked this question of me. First of all, it's primary respiratory. We don't think it's past fecal sewage exposure. It doesn't stay active in the air, by the way. Bright sunlight will even knock down anthrax spores within about 30 minutes. The UV light in the sun and bright sunlight, it does murder. It's just murder on viruses and such. So that can help, but can it survive in a dehydrated state? I haven't seen anybody do studies on that. You'll see a lot of people wearing protective masks that don't protect, and the reason they don't protect is, one, they don't filter properly, and two, most often they're not worn properly. You can't have facial hair and wear a mask and expect it to work. You can't have gaps around the mask. And what's really interesting, you'll see people wearing a mask with no eye protection. Your tear ducts are a major means by which stuff that gets into your eyes gets into your sinuses. Several people have asked whether mosquitoes or other biting insects can spread this virus. Arboviruses are those that are spread by insects, arthropods primarily. And there are 4 genera that are listed there where they can be spread by insects. None of the known arboviruses are coronaviruses. It does not appear that coronaviruses can be spread by mosquitoes or other biting insects, which is good news. So how is it spread? Well, this is for flu, but it would apply here. Turns out that talking, singing, whistling, coughing, sneezing, and breathing can all spread the virus to some extent or another. What's really bad is these hand jet dryers. So if there are microbes circulating around those, or people didn't wash their hands thoroughly and they had the microbes in their hand, they've done studies with flu. These hand jet dryers spread flu virus 1,300 times as efficiently as using a paper towel to dry your hands. I noticed there's a couple of these dryers in some of the restrooms at Monterey, at MIIS. The one that I've used, you spend 5 minutes drying your hands, so eventually you dry your hands in your pants and walk out. But these are not a good thing to be using in a time when there's contagious aerosol diseases floating around. Surgical masks are surgical masks because they're made for surgeons to wear to protect the patient. And the problem with those is that as you breathe into them, they form a perfect location for a virus to survive because it's moist and warm. Of course, the virus can't divide unless it gets into your cells, but the masks are probably not as important in keeping the spread down as a good hygiene. What's wrong with this protection? You notice none of them have glasses on. Your eyes are a primary avenue by which things get into your sinuses. Again, Chinese were actually deep cleaning their cash. A lot of people don't realize it, but American cash, paper money has antimicrobial materials in it, but it's not antiviral. I still don't think that's a major way that this is going to spread around, but I guess it could be in some cases. Again, they've done studies on surfaces. What is important about this is that there's any of a number of household cleaners will kill this stuff. Okay, the alcohol you drink isn't, unless you're drinking something like 130 proof, which I don't think is a good idea, is gonna, with ethanol, you need 71% ethanol minimum. It has to be below 100%. It's got to be 95 below, because for some reason you need some aqueous component. Standard hydrogen peroxide that you get from a store works well, and not only that, but the nice advantage there is that it breaks down to water and oxygen. 0.1% sodium hypochlorite, that's bleach. The bleach you buy is about 6%. Typical, I mean, we used to use it in a BL3 lab. Dilute that 1 to 10, and you get about 0.5%, and that's a very effective to kill the virus. I'm probably running out of time, but I'm going to go a little bit longer here. Basically, any hand sanitizer you use is not going to be as good as regular soap and water because it doesn't stay as a liquid form on your hands as long as it needs to. It will work, but it has to be wet for at least a minute. I would think that your best bet is just to use your normal detergent-based cleaners at home, and you'll have no problems. But right now, they say it can last as long as 9 days on surfaces. The problem with the results they show is they say on shiny, sealed surfaces. Well, when you do the sample, you place the virus on the surface, and then after different periods of time, you take it back off. Well, it's a lot easier to get virus off a smooth sealed surface than it is off a corrugated or rough surface because just the efficiency isn't as good. But still, 9 days max is about all you're going to see. And if you put that out in the sunshine, forget it. It's not going to last anywhere near that long. I'm going to skip ahead a little bit. One of the things you need to do is get people that are sick to stay home. And one of the things one way to do that is when people are hand to mouth and they are sick, they're going to go to work. Most of us have probably gone to work when we probably shouldn't have gone to work. Part of that is because some people are driven by that ethic as I got to get my work done. The other is that people that don't have the money aren't going to take time off if they don't get paid. And therefore, having sick leave turns out to be incredibly important. And this chart is showing you the number of people that different corporations around this country where they work, and the number of people there that do not have paid sick leave. Now, some people say, well, I don't want to do this, I don't want to do that. To me, the paid sick leave is a social thing. It's to protect the rest of the population, not to just provide sick leave for the people that are sick. If you don't want people to go to work when they're sick, then you got to provide them some means to stay home when, especially if they're an hourly. If you don't have that, they're gonna go to work, and you're gonna still have the problems that we're having. There are no vaccines to prevent humans from contracting any coronavirus. There are people working on it. I'm going to flash forward here, but there was never one to develop for SARS-CoV, and the reason for it is that for some reason that nobody understands, that virus petered out after about 6 months, and so the work to develop a vaccine stopped. The only SARS-CoV, the original one, infections after that time were accidental infections with people working on it in a laboratory. The good news is that we know a lot about this virus because we know a lot about SARS, the original SARS, we know a lot about MERS. So there are strategies that have been tried and strategies that haven't worked, some that probably will work. But we do have an advantage in that we know something about how this virus operates. There is no vaccine against the original SARS. What's interesting is MERS started in the Middle East. It's not that contagious, but if you get it, it can be very dangerous. That was in 2012. Well, as of 2017, they thought they had a vaccine ready, but right now there is no current vaccine against MERS-CoV. It hasn't worked. The ones they had going failed. So we don't know what's going to really happen here. When you do develop a vaccine, there's an exploratory stage and then preclinical stage, clinical development, regulatory review and approval, and manufacturing. For the clinical development, there's 3 phases, 1, 2, and 3. The first one, which there's a vaccine in trial for this now, it determines primarily its safety, and they look at antibodies that are made in response to the vaccine and get some idea that whether or not it should go into the next phase. The clinical study is expanded, and vaccine is given to people who have characteristics such as age and physical health similar to those who would normally need the vaccine. And then phase 3 is actually deployment and following the efficacy and safety. There's a Sanofi, I guess it is, became the latest company to jump into this. It hopes to can make it to clinical trials in the next 12 months. That's a long time from now. Other drug companies are doing this. I think 12 months is very optimistic to come in unless somebody comes up with something that I haven't heard of, and there is one possibility there that I'll get to in a second. This Moderna is a US biotech firm, and they have developed a vaccine for trial in just 6 weeks. The first trials are in trial right now up in the Northwest, I believe. And two doses of the vaccine are to be tested at up to 25 volunteers. This is phase 1 testing. Will it work? One thing about this company is their method has never proven to provide a useful vaccine, and it's based on RNA, an RNA technology platform. Now, ideally, it looks like it should work, and their tests say it works, but they've never had a vaccine approved before. You can read through this and how the vaccine is supposed to work. Basically goes in with the RNA that gets expressed in the cell to express a protein that then induces your immune system. But I don't have time to go into the details. The phase 1 trial, again, healthy volunteers are being tested. This is happening in the Northwest right now. And they should have had the first set of injections. They get another one in another couple of weeks. The bad news is that if you need a patent for this, it usually takes 32 months. Normally it takes 5 to 15 years to develop a vaccine. which is then used heavily. And it can take up to 7 years just to get the work done on a regulatory approval, although I think in that case, it'll be, they're going to fast track everything now. There are some drugs that are being tested. Remdesivir is the one that looks like it might be promising, the most promising. It had some impact on MERS in primates. It reduces severity of disease in infected primates. So that drug is, it's called a nucleotide analog, and I'll get to that in just a second. But it was developed initially for Ebola infections, for treating Ebola, and it didn't work for Ebola. But they've continued to test it, and they've been testing it on SARS-CoV-2. Late January, it was administered to the first US patient to be confirmed to be infected in Snohomish County, Washington. It was a compassionate use approval after he progressed into a severe pneumonia with no broad conclusions you can make from this single treatment, but the patient's condition improved dramatically the next day, and he was eventually discharged about a week later. As also in January of this year, Chinese medical researchers started a study with this drug to see if it will in fact work, and that study's in play right now in China and I think in the US now too as well.
Paul?
Yes.
Can I ask you to wrap it up in about 5 minutes? So we have a lot of questions. We have a lot of questions.
I don't answer questions, no. I will easily wrap it up in 5 minutes. I'm almost done. This drug is what's called an adenosine nucleotide analog. What it does is it replaces A in DNA or RNA, in this case RNA, and the A, G, C, and T. It replaces that, and when it goes in, it messes up the replication of the RNA. We don't know if it terminates it at that point or just adds so many mutations that it's no longer functional, but that's how it works. Here are the countries with major activity, more than 800 cases with community spread as of yesterday. And anything in blue means it was reduced from the day before. Anything in red means it increased from the day before. In the case of Italy, it didn't reduce by much. But it gives you an idea of what's going on worldwide. This is the most interesting part to me, though. This in China, you can see the last, well, first of all, March 1 through 15th, they had 836 new cases in that 2 weeks. And now you can see the number of cases over the last few days, well, a week or 2 weeks, basically. And as you can see, the cases are dropping drastically, and most of them are imported cases that they're picking up when people are trying to come back into the country from foreign travel. South Korea, same sort of thing. After 2 weeks, that 2 weeks of March, 4,518 cases, but in the last 2 weeks, the numbers have dropped drastically. It looks like they're beginning to break the transmission of the virus, or it's either that or it's naturally running its course. But it appears that what they're doing is working. Can you catch it from your pets? And these are 3 of my, well, the horse is my wife's horse, not mine. My dog has a hard time understanding the relationship between a dog and a fire hydrant. But it is thought that these, well, first of all, think about the spike protein I talked about. It has to be specific for the species that it infects. And it turns out that SARS-CoV could pass from bats to civet cats. Some people say, oh man, that means your house cat. Civet cats belong to a number of different genera, and none of them are even remotely related to a house cat. House cats belong to the genus Felis. So it's highly unlikely that your cat animal could either get this virus or you could get it from your animal, because it just won't infect those cells. Now, there have been reports of it in dogs, and when they've actually done the studies, it's in homes where there was somebody who was sick, and they detected the RNA from the virus on the dog. Not surprising, because if there's virus around from being shed by the person, there'd be RNA around, and that's what was detected. So anyway, with that, I can answer questions, and I have all afternoon.
Great, thank you very much, Paul. There's a lot of questions. So I'm going to go through the order that's put into the chat window here. And the first question was by Moira. So please identify yourself and give your affiliation. Moira?
Oh, there we go. I had to unmute myself. Yeah, I had a question. You said that the SARS fatality rate went from 2 to 3% to 17% as they had more information, but I would think it would go the other direction because, you know, as you have more information, the denominator of confirmed cases gets bigger.
I think the problem there was that they didn't really do a good collection of people that did not have a severe case of the virus. And so I don't believe that 17%. I think it's probably quite a bit lower than that, but that's what the final official number was.
Okay.
Again, we're doing much more intensive screening now than was ever done there. They didn't have a PCR-based method to detect SARS, the first SARS one.
Okay, and the next question is from Philip. You have 3 questions, so give 2 questions. We'll come back to your third.
Hey, sure. So I had a bunch of questions in the Q&A, but I will just read the first 2 I had. So my questions were, over time, would we expect the virus to mutate into a less lethal form because of natural selection pressures? It's my impression that we see that with similar kinds of illnesses for reasons that you mentioned. And if it mutated too much, I wondered if people acquired immunity, wouldn't that make that acquired immunity less useful? I don't know how much it needs to mutate before that becomes a concern. And the third question is actually related, so I should just throw it out, which is how much do we know about whether people who were sickened have immunity, how much immunity, and for how long? Because my impression is that that actually varies quite a bit.
Okay, for your first question was, let's see, would you repeat the first question? I'm thinking about 3 already.
Yeah, for sure. So the first question was just, would we expect it to mutate into a less lethal form over time?
Yes, yes, and that's because the more lethal form is going to kill the people that infects, and a higher percentage of it, and therefore, and more severe disease, and therefore, though it's going to spread more readily if it's a more mild disease, and therefore the virus, any mutations that allow it to be a more mild disease and spread in the population because people aren't going for medical treatment and such, would mean that yes, you'd expect that, and that's happened in the past. Your second question was, what now?
Over time, if it mutated, would that make acquired immunity, assuming we think people get acquired immunity, less useful?
Probably not, and that's because if it mutates in a way that, okay, the vaccines are going after the spike protein primarily, okay? And that's the protein that allows the virus to access the host cells. So if that mutates too much, it's not going to be able to get into the host cells anymore. And therefore, if it mutates so the structure is enough so that you won't pick it up via a vaccine-based immunity, it's also likely not to be functional to get into the host cells either. Does that make sense?
Yes, it does. And I'll throw out my third related question, which is, how much do we know right now about acquired immunity with this virus? In other words, how much immunity do people have, and for how long do we think they might have it based on either this virus or experience with similar viruses?
Okay, with any immunity, it depends on the response of the body as to what sort of immunity you're going to get. And there are certain cells and certain traits in your immune system that strongly suggest whether it's going to be long-term immunity or not. And in fact, the studies have just come out. There's a couple published in the last couple of days, and it looks very much like this is a normal type of coronavirus in that if you're infected with this, you're likely to be immune from further infection with the same virus permanently. For, you know, it's long-term immunity, at least 7-8 years minimum, but longer than that. And that's based on what they've tracked in people that have had the virus, what changes they're already seeing, and also what they know from MIIS and earlier for SARS. But for MIIS, they know they already have tests for like 7-8 years that people have had MIIS. If they survive, they're immune to it.
So if I can just ask a 2-finger on that one. So what happens when, does the testing quality or the efficiency change when it mutates?
Again, it depends on how they're, okay, if you've been exposed to the virus itself and recover, your immunity isn't just going to be to one protein, it's going to be the whole virus. So even if one part mutated so that it was no longer recognizable by your immune system is responding to multiple factors on the virus, and therefore you'd still, unless all of those changed, which would be virtually impossible, just the probability that is virtually zero, you'd still have immunity to it. But if it's from a specific protein, like the S protein, which would be a vaccine-based one, again, as I said before, if it changed enough so that your immune system didn't recognize it anymore, that would probably change it enough that it could no longer infect your cells either.
But what about the quality of the tests itself? So right now we've developed tests that look for a certain part of the virus, and then does the quality change over time as the virus mutates?
I see. You're talking about the test. Yes,
yeah.
It could. But the fact is that as these things come out, they're getting sequenced really fast, and it takes you could change the sequence of your primers that you use in your test very quickly, just to keep up with any change in the virus.
Great. Okay, thank you very much. So the next question is from Isaac. Please identify yourself and also give your affiliation.
I'm a student in the non-proliferation and terrorism studies. I've heard a lot of news that the coming months, as it gets hotter, that it might help, I wouldn't say defeat the virus, but at least make it less severe. Is that true, or is that not true?
Well, I can give you two answers. First of all, MIIS is in Saudi Arabia and other places like that, and it seems to do just fine. And last I heard, it gets pretty hot over there. The other side of that, though, is probably not about heat. It's more about sunlight. Even anthrax spores don't survive more than a short time, 30 minutes to an hour in bright sunlight. Once they get in the soil, they last for decades. But if they're out in the sun, they don't last very long. And the same thing for viruses. They're getting hits with the UV. And remember that this virus, when it's out in the environment, is not in a host. It's floating around out there. And therefore, there's no way it can repair any damage that's done. So you get enough hits on it, on the genome with UV light and on the proteins with UV light, it's going to kill it really rather quickly. It's easy enough to do tests. We used to use UV lights in our hoods in our BL2 labs, and you put materials in there that are actually, you start with something that's very resistant to the UV, and if it kills that, you assume it killed everything else. And believe me, UV light is a very good disinfectant, and sunlight is, if you've ever had a sunburn, you know what it can do in a fairly short period of time. So yes, I'd expect it to drop down, but we don't know whether it might reoccur in the fall, if it become a seasonal disease or something else.
Okay, thank you.
Great. So the next question from Nomza, please, you have the floor.
Hi, Dr. Jackson. I just had a question about why it seems to be so infectious and spreading so quickly to be causing a global pandemic.
Well, it appears to have come, as we said before, out of an animal population. Why is it spreading as quickly? It has to do more with the cells that are first infected, and those are the cells in your upper sinuses. And therefore, once those are infected and they start to shed virus, you're shedding a lot more out of your system than, let's say, if they were down in some other tissue in your body, because your sinuses, I mean, if you've ever seen actual videos of it with a black background lighting, talking and breathing actually puts out quite a bit of mist from you as you speak or breathe, let alone coughing, sneezing, singing, whistling, all that sort of thing puts out an aerosol into the air. And so that's one of the reasons it's more contagious. The other is that it seems to be very infectious. that when you are exposed, it does not appear to take that many of the viruses to cause an infection. So for example, something like anthrax spores, it takes 8 to 10,000 spores to get into your lungs to cause an aerosol infection. Tularemia, different bacterium, takes 1. Plague takes about 5. So it's a matter of how fast and how efficiently they can establish an infection in the host as to how fast it spreads and how many people get sick.
Great, thank you. Mark Bishop, do you have a question as well?
Hi, Paul. You may have, excuse me, haven't used my voice yet today, sheltering in place.
Stay home.
Anyway, you may have already answered this, but I got distracted by my son dropping by to get tools to install his bidet.
Okay.
But my
Hey, did you say
questions is not about hoarding toilet paper.
Okay.
So I read somewhere today that the virus is not mutating significantly, which is good news for the possibility of a single vaccine giving lasting protection. And do you know if this is true? And if so, why would this be with an RNA virus?
Well, it's not going to mutate as fast as flu because it only has one factor involved, and that is that it doesn't, it's the mutation rate only that determines the change in the virus. Flu and some other viruses, mostly flu though, the reason that you have to have a new vaccine every year is because not only does it mutate, but instead of having one long piece of RNA, it has 8 individual pieces of RNA. So what happens is that 2 different strains of flu might infect a host. If they get into the same, if those 2 different viruses get into the same cell, all that has to come out to be a viable virus progeny is one that has 8 segments, all 8 of one of each of the 8 segments. It doesn't matter whether it came from parent A or parent B. And so not only do you have mutations occurring, but you have this rearranging going on and it's called reassortment going on all the time. So the chances of much more variability in the flu virus population is much, much higher than it would be for something like this. On top of that, there are factors we don't understand. It may be in the infection that the mutations are occurring, but that only a narrow range of them can survive in the host and be released. So we don't know that. But there is that possibility that what's happening is there's only a narrow range where this can survive in humans. And any mutations that take it out of that range are lethal and it's taken out of the mix. But again, we don't have the answer for sure.
Thank you.
Thank you, Paul. Are there any more questions? I think at this point, just speak.
Frank?
Yeah, go ahead.
If we want to share the recording of this webinar with other people, where can we get a link to that recording?
So I'm recording it right now and I'll send you the link.
Okay.
And we'll make it available.
Okay, are some of these people the students from the class?
I'm in Professor Blake's class. It's me, Dr. Jackson Reyna.
Okay, because I'm going to be sending out slides for the class, and this lecture, if you will actually probably be 2 full lectures, so like 4 hours. There's going to be a lot more detail presented in the class, and if at that time, friends or others, you want to record it, we can have it recorded then too. And this is fully distributable to anybody who wants it.
Great, that's really great. And also, I believe that maybe in the next few weeks, when our class starts, then you know, a lot will change too.
Well, that's just it. I updated this morning actually, and I don't know if you remember the one I sent last week. It was a lot longer, and then I shortened it down to 60, and then I'm back up to 93 slides again, or something like that.
Yeah.
It's just every time you turn around, there's new information.
Yep, I totally understand. Okay, I think thanks a lot for the presentation. I don't know how to virtually clap here, but I'd like to clap.
If everybody went on mute, we can all clap.
Well, it's okay. Clapping is understood.